we have time for a few questions, and we are going to start session 2 on time at 9:10. So who has a question for either Dr. Quadrato, Dr. Tang, or Miss, I know I butchered your name. You're going to say it.
Kostyanovskaya. Sorry.
Yeah, I said Skaya, but it's Oska. Okay. So yeah, who loved that? Wasn't that amazing? Oh, here's a question over here.
Right. So this so I showed in the PKBD study where you can do either oral gavage or through the IP. Yeah, but so that shows it's already available, which is similar to the clinical trial. So that was delivered through oral. But in the mouse, it's actually much easier to do injection than doing the gavage. So most of the efficacy study we did IP injection. Yeah, yeah. Good.
Sure if it was on or not. Really nice work. I have a couple of questions about this kinase inhibitor because I've seen it before and implicated for other neurodevelopmental disorders. Have you looked at the gene expression changes that occur as a result of treatment with this kinase inhibitor?
Yes. We have
it's pretty pleiotropic, right?
Yeah, we have bioarchive paper that's online, so Cantabile can send you the links. So we did a lot of transcriptomics analysis.
Yeah.
So we know the kind of yeah.
So how many genes are dysregulated?
That's a good question. So this response, what we found, it's about 700 in both mouse and the human neurons. About 700 gene expression would go up, and about 2,000 gene would go down.
Exactly.
And there's a pattern there. So genes expression going up, a lot has to do with neuronal maturation. So it's a lot of these synapse genes, a lot of transporter ion channel type of genes expression go up. The genes that expression go down, most has to do with neuroinflammation. So it's a completely different categories of genes regulated by this kinase. So really, we are trying to understand this biology, understanding this, and how to match the patients with the drug that they benefit from. Yeah. Good.
Yeah, I had a question for all the speakers. Each of your presentations highlight potentially a GABAergic phenotype. So can you guys comment on the striatal phenotype for speaker 2, the PV phenotype for speaker 3, and then for the forebrain phenotype, do you see defects in the inner neurons?
Interneurons. Yeah.
So we have been focused on really studying excitatory neurons. But basically by looking, this is for the forebrain part. I think, by looking at expression of Syngap1, I think there is actually expression also in interneural. And we have this general idea that actually Syngap may have a very ubiquitous function in most of the neural cell types, so including astrocytes and interneurons. So we haven't really done a thorough characterization, but I think I'm expecting problems also in interneurons. And when we look at association of Syngap1 expression in enteric neurons, actually it seems like the highest level of expression are in inhibitory neurons in the enteric nervous system. So I think it will play definitely a very important role there.
Echoing the same sentiment, we haven't looked at specific neurons and the expression of Syngap1 at cilia and specific neurons yet. That's sort of future work. But there's already work in review, I think from the Hugenir lab, that talks about different isoforms being important for excitatory synapses versus non-excitatory synapses. But another thing I want to mention that's sort of along these lines is that they've actually shown that, like, for example, knockdown of cilia in neurons in general, I think these are projection neurons, specifically results in dysfunction of only excitatory synapses, but the inhibitory, so GABAergic synapses are fine. So for that reason, and because of the importance of excitatory neurons in autism from Jeremy Woolsey's lab, we focus on excitatory cortical neurons so far.
Yes, so my lab actually does a lot of work in the GABA inhibition space, right? So this what I showed is this F83 kinase expressed pretty much in all neurons, but it's higher in the interneurons. So it's just look at expression level. So this is we have staining results, and also the transcriptomics looks the same. But this, but really, I want to point out one thing is about the GABA inhibition. So we study a lot on the KCC2, that's chloride transporter, controls the GABA response in all neurons, excited and inhibiting neuron. And we have a lot of, I think the KCC2 and Syngap is actually a lot of this kind of synergy or some sort of, at least some association study. It seems like if you reduce Syngap, the KCC2 as a result of the efficacy of GABA inhibition will also be impaired. So I think there's this rich space, how the inhibition is impaired in this genetic disorders. Yeah.
Question right here. Question right here.
My question's for, can you hear me? My question's for Dr. Tang. So with the increase of SYNGAP1 expression that you're seeing with the drug that y'all are working on, how are you anticipating this works for missense mutations and where there's SYNGAP1 seemingly present already, but not really effective? And how are you anticipating, or are you considering how effective that could be for that whole group of patients?
That's an excellent question. So I showed a kind of simplistic diagram that there's two assembly lines and one get decommissioned, but there could be scenarios. So that works for this called a truncated mutation or nonsense mutation, right? So one is really mutated, but for the missense, basically it's still producing, but maybe it's a defective part. So then really it's a couple of scenarios. One is that parts gets produced and get degraded. Right, so the cell somehow can sense that didn't pass quality control, right? So another possibility is that defective part can jam up the good parts, right? Jam up, so that's called dominant negative. So really, that's important to really understand this what's the nature of this variants, right? Well, which part, which are nonsense, which are kind of loss of function, but which are dominant negative. In our case, the drug work on both alleles. Looks like it work on both alleles of the gene, right? So then we basically show that in the KCA2 really. You work on both allele gene. In that case, you really want to target this either nonsense mutation or like a loss of function, but want to avoid the dominant negative, right? So then that's really so need to be kind of case by case, need to analyze the biology. Yeah. Okay. Yeah.
Okay, I have a question for Elina about cilia, and it's great to see the expression and it's great to see the nasal nitric oxide. And I was wondering about in vitro cell assays and what you're doing to see with mutant SYNGAP if there's mutant cilia function. One thing is with the beating cilia on the surface of the brain, if that's making CSF not move around, that's pretty interesting. And if that's something that needs to be fixed in order to have ASOs get around, there's lots of reasons why we'd want to know this, but that's one that comes to mind.
So I'm actually going to start with motile cilia since you just ended on that. So recent work actually showed, I told a small little the tiniest white lie, which is that up until very recently, it was thought that the beating cilia were important for moving cerebrospinal fluid. But actually a lab showed quite recently, I think it's on bioRxiv now, that in zebrafish, if you get rid of beating cilia, the cerebrospinal fluid moves more or less fine, like within a small margin. But the real issue is that the excitatory to inhibitory balance in the brain is now messed up. And so in zebrafish, one of the things that they do, which we can do similarly, is you can actually measure neuronal signaling in real time using a calcium sensor and actually do in vivo live imaging once you do this sort of knockdown. And so that's one of the things that I'm personally interested in, is looking at ablating cilia at really specific times or places and seeing how that affects neuronal signaling. But you can also do these sensors that work within a cilium to see, look at secondary messengers like calcium and cyclic AMP and see how those change when you look in a SYNGAP1 variant. And the last thing is we can also look at ciliary beating live using some really cool microscopy techniques kind of specific to frogs. So hopefully that answers your question. We can look in vitro in a dish using some signaling sensors, but I'm really excited about in vivo work because it's kind of about how it all comes together for a real functional brain. So.
That's excellent. Thank you. Okay, let's give a round of applause to our speakers. And if you guys can exit that way, we have some speaker gifts for you. Marta and Julie will do speaker gifts over there.